Audrey, and thanks everyone for coming out this morning. So I can advance with this. That's what I'll do. All right, so thanks everyone. I'm going to talk a little bit about medications in milk and primarily focus on the UC San Diego Human Milk Research Biorepository, which is a core resource that has been the focus of our activities in the MPRINT initiative. So if you're not familiar with this, this is a resource that was established in 2014, so it's about 12 years old now, and it involves recruitment of women who are breastfeeding infants or toddlers of any age who reside in the US and Canada. So it's a broad catchment area for enrollment, and it involves consenting participants to provide breast milk samples, and we primarily use a home collection method, so this is novel. The methods have been developed and are being used by some other milk biobanks in other parts of the world, but still a work in process. And the reason we did that and continue to do this is obviously it gives us the opportunity to recruit a much more broad array of people with different kinds of medication exposures and other characteristics, some of which are quite rare, by extending the catchment area beyond a local clinical site. So everyone who enrolls provides a sample and associated metadata. So close to the time that they are providing the sample, they engage in a telephone interview where they provide information about demographics and their health history, child's health history, the characteristics about their breastfeeding experiences and the sample collection. And then they do a 14-day exposure history day by day of every medication, vaccine, dietary supplement, herbal product, recreational substance that they've used day by day, dose and indication. And they complete an infant adverse reaction checklist. They complete stress, anxiety, depression questionnaires, a physical activity and food frequency questionnaire, and then relevant to the age of the child who's being breastfed, they complete a series of neurodevelopmental questionnaires, and then they give us permission to extract data from their medical records, so we capture the medical record for the mother and the child wherever they live. And optional in the consent is a genetic consent, so the permission to be able to use the samples for genetic purposes. The sample collection is just briefly described here, but basically we have a whole procedure outlined for the participant and instructions on how to collect the sample. We send them a kit, how they collect it, how they briefly store it in the refrigerator, send it on ice packs overnight via FedEx, where we are alerted, receive it, process it, aliquot it, and put it in the biobank. And the method has worked successfully over the last 11 years. So we've also recently expanded our lab capacity for processing and storage. We started out with shared space through our CTSA, which we grew out of, and it became cost prohibitive. So we built out in our own space lab area. We now have 10 freezers in there and some room for expansion, and then have the equipment and space to do other types of processing in addition to aliquoting the samples. We have recently revamped our website, and this is now a much more user-friendly resource for researchers. So in addition to all the data about why enroll, what kinds of activities does the Human Milk Biorepository engage in, it has a data platform now which gives basically pretty real time, it's updated weekly now, information on what's in the biorepository and the characteristics of the samples that are there, because this is intended to be a research resource that others can contact us about being able to access. So as you can see here, as of April 2nd, there's about 3,500 women enrolled in the study. study, about 8,000 milk samples and 122,000 aliquots. And then, as you can see on the right, you can filter on the website for any of the characteristics that you're interested in and see how many samples are available that meet your criteria. On the dashboard, you can look at the distribution of the samples that are in the overall cohort, and then again, you can select by income, race, ethnicity, maternal age, education level, a variety of characteristics, and then we have a drug dictionary in here. So you can scroll down and select specific medications or groups of medications and see how many unique women with samples we have in the biorepository. So we think this is really going to make the utility of the biorepository much more readily accessible. So in terms of our MPRINT research activities, our project in the HMB was a clinical project focused on trying to understand better what impact, if any, there is of a mother taking antibiotics while she is breastfeeding on the infant in terms of a variety of different outcomes. And so the design was that we wanted to be able to collect samples and antibiotic exposed, so mothers who were taking antibiotics from the beginning of treatment to the end. But the best way to do that was to try to build a sort of a baseline group of moms and then follow them until they got a prescription for an antibiotic. So we did recruit 480 mothers through the Human Milk Biorepository and consented them to participate in the study. We collected baseline milk, skin, stool, and environmental samples from all of them, and then followed them to see if, in fact, they started taking an antibiotic in the subsequent months. And the target was to identify 40 breastfeeding mothers who continue to breastfeed who started an antibiotic. So we did that, nested in the cohort. We've identified 40, and at the same time identified age and sex-matched infants whose mothers were breastfeeding from that same baseline cohort who did not start an antibiotic. And in both of those groups, we collected serial samples, milk, skin, stool, and environmental samples. So the sort of first pass at looking at this was just to see, were there any significant differences between those two groups overall on infant growth measures, the incidence of serious or non-serious infections in the children, or performance on a screening for developmental milestones, the Ages and Stages questionnaire, which is a maternal report at 12 months of age. And we did not see significant differences, but the really interesting findings I think you're going to hear about in the next talk. Dr. Sonoda is going to go over the additional analyses that were performed. And Dr. Momper did PK on the samples, and then Dr. Sonoda's lab worked on the metabolome and microbiome components of this. So more to follow. And then our hope is that we will continue to follow up these children through 24 months with an expanded set of neurodevelopmental questionnaires that involve screening for autism and language development and repeated measures on growth. So with supplemental funding, we also were able to enrich the value of the cohort by taking what at the time was everyone in the biorepository, 2,500 bank samples from unique individuals, and Dr. Sonoda was able to utilize those for untargeted metabolomics, and a very rich data set, which I think you'll hear about in the next talk. And then with the milk analytics core led by Dr. Bodie, we recruited some new women and had archive samples for 600 individuals who had either taken a monoclonal antibody or an SSRI, and those were assayed for human milk oligosaccharides, and that data is available. And then we did add, it as part of a supplement, it tested whether or not it was feasible to collect dried blood spots from mothers and infants in this mailed home collection scheme. So we added Tasso kits to the consent process and sent them to the mothers with instructions on how to collect it from themselves and from their infant. And these little tiny kits, I thought they would not do it for the infant. They might do it for themselves, and it turned out it was highly successful, and more than 90% agreed to do it and were able to get the kit collection completed for themselves and their infant. So for a subset, we have those now. We've had a number of collaborations built as part of MPRINT, and one of them, this was initially funded by NICHD R21, was to try to better understand whether this home-based collection of milk samples in any important ways differed in the components or composition of the samples compared to clinic-based sampling. So can we compare these apples to apples? And so we are still in the process of doing some additional data analysis on this, but the first paper came out, and Dr. Kathleen Jobe, who presented a couple days ago at this meeting, is the first author on this, comparing mimicked home-based and clinic-based sampling strategies for breast milk and blood collection. And I think where it's going to land is that it's comparable for some drugs and for drugs that have maybe more sensitive stability profiles, it may be less comparable. So that more to come on that. We've also collaborated with the Pediatric Trials Network, and this is in collaboration with Dr. Elizabeth Thompson, who presented these data a couple of days ago at this meeting. So the question here was, we asked the question, well, we are collecting samples already in the Human Milk Biorepository and have quite a few there that are with medication exposures that are targeted medications for the CUDDLE study. And the CUDDLE study, as you're probably aware, is a NICHD funded study that's been ongoing, targeting various selected drugs of interest, where a relatively modest sample size is being collected in various clinical sites under strict conditions to try to better understand how much drug is in milk and various other characteristics, so that that information can be included in the product label. So we asked the question whether or not the home collected samples we had for those same exposures could be combined with the CUDDLE study samples, increasing the robustness of the data and ability to extend information about longer term child age exposures for inclusion in the product label. So would it be comparable? So as Dr. Thompson presented, what she did was look at through the CUDDLE study, mothers and infants with sertraline exposure, where they had paired samples for milk and maternal and infant plasma. And by design in that study, they had to be recruited prior to 6 months of age. And then from the Human Milk Biorepository, we had 198 mothers with sertraline exposure with human milk and some with maternal dried blood spots, and they were recruited in the first 24 months of age. And so the data that she presented, obviously much more extensive than what I'm saying here, but I think basically the overall preliminary conclusion is that there are not huge differences between the two sources of data. There's huge variability in the sertraline levels in milk by age and by individual, but there may be some comparability between these two sources of sample collection that would make it beneficial for inclusion in the product label. So more to come on that. And then the last collaboration was with the Vanderbilt MPRINT Center of Excellence, and what we wanted to do was first demonstrate that yes, you can genotype mothers through milk, so extracting DNA from milk. And then we wanted to look at whether the drug metabolizing enzyme genotypes of the mothers corresponded with drug levels. And in this example, again, sertraline, 50 samples from the breast milk biorepository. The drug concentrations were measured by Dr. Momper in our Center of Excellence. And then the Vanderbilt group is just doing the analysis of the genotyping data, and preliminarily for CYP2C19, you can see here the distribution in terms of the phenotype for poor to ultra rapid metabolizer. And as you look at the range for the drug concentrations for sertraline on the right side, you can see there's not just a super direct correlation here. And so again, a lot of variability in drug levels, even given the same dose in the mothers in the milk. So more to be done here with the other drug metabolizing enzymes. So a few recent findings, working with the core data from the Human Milk Biorepository. These are a few papers that have been published in the last year with our collaborators in the Center of Excellence, and also with a visiting scholar, a neonatologist, postdoctoral fellow with us from the Karolinska Institute. So the first one was addressing this kind of age-old question of do women, for whatever reason, due to fear that a drug may not be safe to use while breastfeeding, or lack of information that mothers may decline to breastfeed, they may not breastfeed as often, or they may discontinue breastfeeding earlier than recommendations. And so what in this paper, what Dr. Heinonen did was look at anti-inflammatory drugs, antidepressants, and asthma drugs as a class, and looked at whether women either did or did not initiate breastfeeding in those classes, whether they supplemented with formula, and then she looked at whether they had stopped taking their medication during pregnancy or continued during pregnancy, and how that related to breastfeeding or use of supplements. And as you can see, there were some differences in anti-inflammatory drugs, that women taking those drugs were much less likely to initiate breastfeeding, whereas with antidepressants and asthma drugs, not a significant difference there compared to people who were not exposed. And then those who discontinued their medication during pregnancy, in terms of breastfeeding, again, anti-inflammatory drugs stood out as a risk for not breastfeeding. Same kinds of findings in terms of duration of breastfeeding time in the first 6 months postpartum. So suggesting, and this is speculation, that perhaps for antidepressants, people feel more comfortable with more years of data in terms of initiating breastfeeding, whereas with anti-inflammatory drugs, for many of them, far fewer data available. The second analysis, and this is, I know Dr. Sonoda will be pursuing the idea that it's really important not only to look at drug levels in milk to say, it's there, or it's there at less than 10% or whatever, but to look beyond that in terms of medication use during lactation, and whether or not it has any effect on milk composition, other aspects of milk or breastfeeding. So in this analysis, what Dr. Heinonen looked at was macronutrients in milk, and again, looking at antidepressants as a class and anti-inflammatory medications, and this was just published last year. So the SSRIs, there were 63 in that sample, monoclonal antibodies, a small number of systemic steroid users, other anti-inflammatory drugs, disease-matched controls, and then healthy mothers that were matched by age and sex of the infant. And there were differences she looked at carbohydrates, fat, protein, and total energy, and found that mothers treated with anti-rheumatic medications and SSRIs on average had lower levels of protein in their milk samples compared to disease-matched unmedicated mothers, and mothers treated with anti-rheumatic medications. Other anti-rheumatic medications contain lower levels of fat and total energy than samples from disease-matched unmedicated mothers. Whether or not this is clinically significant is undetermined. The findings were only to say that there are differences, and that this is something that deserves further study. And then third, working with Dr. Bodie's lab and the human milk oligosaccharide assays that I mentioned had been done previously, looked at, is there variation in human milk oligosaccharides in mothers treated with antidepressants and anti-inflammatories? And this was just published in Pediatric Research. And she looked at overall relative abundance, and looked at it in secretors and non-secretors, and did find that there were differences in antidepressants and anti-inflammatory drugs versus the unexposed, and found that there were differences in secretors and non-secretors for individual HMOs in the antidepressant or anti-inflammatory drug-treated mothers compared to disease-matched controls. Again, is this clinically significant? that was not tested in this study, just raising the question that this is something that deserves further study. And then last, this is, when this went out for review, the comment was, well, duh, we already know this, but we really don't know this because there hadn't been any data published on this. This is looking at cognitive outcomes of children exposed to SSRIs through breast milk. So the sample is women who had been taking SSRIs during pregnancy and then either breastfed while taking SSRIs, breastfed not taking SSRIs, or didn't breastfeed. And the children were followed to about 4 years of age and tested using the Wechsler preschool and primary scales of intelligence. So this is just looking at IQ, samples small, 22 exposed during breastfeeding, 37 not breastfed but not exposed to an SSRI during breastfeeding, and 38 not breastfed. And you can see, there are mean differences, but the confidence intervals overlap on full-scale IQ, verbal IQ, and performance IQ. So although of the 97 children whose mothers were treated with SSRIs during pregnancy, the breastfed children on average had 6 to 8 points higher full-scale IQ at 4 years of age. But again, the differences between the groups after adjustment for important factors was not statistically significant, and maternal depressive symptoms during pregnancy were strongly correlated with child IQ, but suggesting overall that in terms of a longer-term outcome for mothers who breastfeed while taking SSRIs, absolutely no indication that there is harm in terms of child cognition. So I obviously want to thank NICHD and the MPRINT team for support of the work that we've done, the NIEHS for infrastructural support for the biorepository, and NCATS, our CTSA, has provided some support, and also Carrie Bertrand, who is the research manager extraordinaire who runs the biorepository, and Essie Heinonen, who is the neonatologist who worked on all of these studies I just described over the last year. So thank you, and I think we're going to hold questions till the end, so I'm going to introduce Dr. Tsunoda.